Hello students, welcome back. This lecture will be Chemistry of Life, Part 6. We will continue with our 4 macromolecules, the proteins, the nucleic acids, the carbohydrates, and the lipids. We've already covered the proteins, and so we will, hopefully in this lecture, we will complete the other 3. The Chemistry of Life, Part 6. There are 4 macromolecules in the human body, or in human cells. We came up with a mnemonic for those: pencil, P-N-C-L. P-N-C and L. P is for proteins. Proteins are polymers of amino acids. And so you have a lot of amino acids make a long string of proteins. Nucleic acids are strings or long polymers of nucleotides. Carbohydrates are long strings of sugars. And lipids don't make polymers, so we are going to talk about lipids in this context. Now, so in general, we have 3 polymers, and so of those 3 polymers, this would be the general term. And those polymers, the individual units of those polymers are called monomers. I'm going to put here a monomer. M-O-N-O-M-E-R. And so you have lots of monomers joined together to make a long string. And that long string is called a polymer. So in the first example here, the protein is the polymer. The monomers are amino acids. In the second one, the nucleic acid is the polymer, and the monomers are the nucleotides. In the third example, the carbohydrate is the polymer, and the monomers are the sugars. To make I will talk about this later, but just as an introduction, to make a polymer, what you have to do is take away water. So if you have another amino acid here and you want to join this amino acid to the long string, what you'll do is you will remove water, and that process is called dehydration synthesis, because you're synthesizing the polymer by removing water. Dehydration synthesis. And we'll talk about this more later after we've talked about each of the polymers. And to actually break a polymer, we actually have to add water. And so when you add water to shorten a polymer, we call this process hydrolysis. So hydro for water, we're using water to lyse the polymer. Hydrolysis, or simply hydrolysis. Okay, and so, but the main point of this slide here is just to show you each of these 3 macromolecules, each one is a polymer, and each one is comprised of numerous monomers. In the case of the protein, the monomers are amino acids. In the case of the nucleic acids, the monomers are nucleotides. In the case of carbohydrates, the monomers are sugars. Okay. And to lengthen the polymer, you have to remove water, and that process is called dehydration synthesis. And to shorten the polymer, you have to add water, and that process is called hydrolysis, water splitting. or hydrolysis. And that's all I'll say about polymers and polymerization. The next polymer that we will look at will be our nucleic acids. There are two primary nucleic acids, DNA and RNA. So let's start with DNA. DNA is kind of a doubled helix. Okay, that's one strand of DNA, and let's draw a second strand of DNA. Okay, now on DNA there are nucleotides, and these nucleotides, there are 4 nucleotides. I'm going to make this really simple here. Okay, there's a G here. There's an A here. These are your big ones. G and A are the long ones. And then there's a short one here, a T. Then there's another short one, C. I'm going to make this as simple as oops, I'm sorry, I did something wrong here. Actually, I want to put all of this on my left strand, on my red strand. These are going to be short, T, and another short one here, C. Okay, so we have 2 long ones, G and A, and 2 short ones, T and C. And so those will pair up. Let's make these green. So the G is going to pair up with a short C, and the A is going to pair up with a short T, and this T, this short T, is going to pair up with a long A, and this C is going to pair up with a long G. Okay, and there's DNA, and DNA is a double helix. Now between these we're going to have some hydrogen bonding. Let's see here. I guess a good color for hydrogen bonding. Let's get like a purple color. Now between G and C, the hydrogen bonds are going to always be, there are always 3 hydrogen bonds holding them together. And between A and T, there are 2 hydrogen bonds. Okay, there are 2 hydrogen bonds here, and between C and G there are 3 hydrogen bonds, and that's DNA basically. Now, the backbone, this backbone of DNA here, I'm only going to show one of them. This backbone of DNA is composed of 2 components. Okay. One of them is, we're just going to put an S for sugar, and the other is going to be a P for phosphate. And so then it goes down here. We're going to have another phosphate and another sugar. And so these 3 components together, the phosphate, the sugar, and the A, or the nitrogenous base, those 3 things together. I guess I could highlight this. these three components together. These three components together, these make up the nucleotide. Okay, and so all three of these together make up the nucleotide. L-E-O, nucleotide. Now the individual letters here, like G, A, T, and C, each of them is a nitrogenous base. So a nucleotide is made up of a nitrogenous base, a sugar, and a phosphate. And so these individual components here, this is called a nitrogenous base. There are 4 nitrogenous bases. There are 4 nitrogenous bases. There are the big ones like G and A. They are called purines, and these are purines. You can remember pure as gold. A-G. Adenine and guanine. These are our purine, and those are the big ones. And the small ones, these are called pyrimidines. And they are cytosine, and they both have Y's in them, so that'll make them easy to remember. Cytosine and thymine. Okay, and this is DNA, and when we come back, we'll start with this slide, and we'll talk about some other components of DNA here. The structure shown here is the molecule. It is a nucleic acid, but specifically it is DNA. So we have shown here DNA. This is DNA, or deoxyribonucleic acid. Now the purple bonds between the nitrogenous bases, those are hydrogen bonds. These are hydrogen bonds. So these are hydrogen bonds shown between the nitrogenous bases. There are 4 nitrogenous bases shown here. Adenine, guanine, those are the purines, those are the large ones, and 2 pyrimidines, the smaller ones, cytosine and thymine. Now there's one other nitrogenous base, and these go in the second type of nucleic acid, and that's going to be RNA. And so let us draw an RNA molecule. So let's get rid of DNA for now, and let's draw RNA. Now RNA is just one single strand, and most of the time it doesn't have any folding. And so for RNA, you're going to have, again, your 4 nucleotides. You're going to have your let's do it this way. You're going to have 4 nitrogenous bases. G, which is a big one. A, which is a big one. C, which is a small one, it's shorter. It's a pyrimidine, and you don't have T, but in place of T you have another short one, which is another pyrimidine, uracil. So this is going to be RNA. And RNA is the second type of the second type of nucleic acid. And you have 4 nitrogenous bases, G and A, which are our purines, just as in DNA. But in RNA you have U instead of T. So just so you remember this, I'm going to put a T here. And what we'll do, we'll put a big X through it just to show you that there is no thymine in RNA. And just as before, in this one, we're going to have the backbone is going to be composed of a phosphate and a sugar. A phosphate and a sugar. And so that's going to go all the way down. All of that's the same. Now, the sugar here, the sugar in RNA, that sugar is called ribose. And we are going to learn the structure. And that's why this nucleic acid is called ribonucleic acid, because the sugar is ribose. And let's just go back to DNA just for a moment. The sugar in DNA let's see here. The sugar in DNA is called deoxyribose. And that's why this sugar is called, that's why the nucleic acid is called deoxyribonucleic acid. So that's it for DNA, and this is it for RNA. Okay, I'm going to erase this. Sorry about that. I'm going to erase this here. We don't need this here. And so this is RNA. RNA is a single strand. DNA is a double strand. RNA has ribose. DNA has deoxyribose. RNA has uracil. DNA has thymine. And that is all we will say about the nucleic acids. And just remember, the monomers in nucleic acids are nucleotides. Carbohydrates are polymers of sugars. I'm going to draw for you 3 sugars that you should know the structure of. Okay, this first sugar is a ring, is a 6-membered ring. Okay, there's our 6-membered ring for this sugar. Okay, and on one of these, on this carbon here, we have a CH2OH group. Now on the rest of the carbons, we have an OH group that could go either up or down. Here, the OH is down. This one is down. Now this first one here doesn't matter if this one goes up or down. So this is, we can call this carbon 1. This is carbon 2. Carbon 3, the OH is going to go up. Carbon 4, the OH is going to go down. So this is 3, 4, carbon 5, and carbon 6. Anywhere else where there's not a double bond, anywhere else where there's not a full complement of 4 bonds on the carbon, it's understood that there's a hydrogen there. So these hydrogens are understood. So that's one way to draw this structure. So the way that I remember it is, it doesn't matter if this is just starting at carbon 2, down, up, or you could put the first one up, 1 up, down, up. It doesn't matter with that first one. So this sugar, the most popular of all the sugars, is glucose. Now there is a simpler way to draw glucose. What we're going to do this time, we're going to draw the glucose, but we aren't going to draw we aren't going to show the carbons explicitly in it. We aren't going to show the hydrogens. They're going to be understood. So let's draw our oxygen here. CH2OH. And again, this first carbon, it doesn't matter. The first OH, it doesn't matter if it goes up or down. So we'll just put it up. And we aren't going to show the hydrogens. And this one is going to go down, up, and down. So the way I remember glucose is up, down, up, down, up. Everything is just the opposite as you move along. They're just opposites of one another. And so this sugar is glucose. The most common of all the sugars. That's glucose. Let's draw another sugar. Okay. We're not going to actually draw it now. But when we come back, we're going to look at another sugar that's very closely related to glucose. And so that's the structure of glucose. That's the one that I'll need you to know. Let's look at the next sugar. It's very similar to glucose. Very similar to glucose. Let's get rid of this first glucose. Let's get rid of this one. We don't need it. Let's look at the simple glucose. So let's move it over here. And let's just make ourselves a copy of this one. So we're going to make a copy of glucose and move it over here. And let's change this sugar in some kind of way. Okay, the only thing we have to do to this sugar to turn it into another sugar is change this carbon here. This is going to be carbon number 4. So this is carbon 1. I guess I should label these. So we have carbon 1, 2, 3, 4, 5, 6. And so we're going to change the orientation of this hydroxyl group on carbon number 4. Okay, so let's do that. Zoom in here. Let's get rid of this OH group. Okay, very good. Now, let's put the OH in the up position. OH. Okay. And that's all we have to do. And now we have a brand new sugar. The sugar is no longer glucose. So let's get rid of this. This sugar is now called galactose. G-A-C-T-O-S-E. This sugar is galactose. And the only difference between it and glucose is here. Carbon number 12, carbon number 4, this OH is up. Okay. And so on the left we have glucose, on the right we have galactose. And there's only one other sugar, one other simple sugar that I'll need you to know. We'll look at that. It's kind of completely different than these two, and we'll look at it on the next slide. The next sugar that we're going to explore, it's still going to have 6 carbons, but it's just going to make a 5-membered ring. Let's just put the outline of the sugar first. Okay. Just going to be a 5-membered ring here. There it is. This doesn't look pretty good. Okay. Now let's put all of our carbons on here. Our other carbons, rather. We're going to have a CH2OH here. And we're going to have a CH2OH here. And so these are our 6, we have our 6 carbons here. Now let's put our OHs, our hydroxyl groups. We'll have an OH here, going up, an OH group here, going up, an OH group here, going down. And so that gives us our 3 lone hydroxyl groups. And so as you can see from this sugar, we have 6 carbons, we have 12 hydrogens, and we have 6 oxygens. And so this structure has the same molecular formula as that of glucose and galactose. This is called fruit sugar, or fructose. The sugar is called fructose. Okay. And this is the third simple sugar that I need you to know. Now, each of these sugars is called a simple sugar. And they're also referred to as monosaccharides. Mono meaning one, saccharide meaning sugar, ide, a monosaccharide. Sometimes you can put 2 of these together. For example, if you join I'm not going to show the structure here, but if you join glucose, we'll just put GLC for glucose, and glucose together, we call that maltose. And ose means sugar or sweet, just like saccharide means sugar or sweet. If you put glucose and galactose together, we call this lactose, or milk sugar, the sugar that's in milk. And lastly, if you put glucose and fructose together, we call that sucrose. And sucrose is just simply table sugar. Lactose is milk sugar. I don't know if I can fit that in there. Maybe if I move this down. Let's see if I can move this down here. Okay, perfect, perfect. Now I will be able to put here lactose is just milk sugar, or the sugar that's in milk. Okay, our three simple sugars that we should know. Glucose is good. It would be good to know this structure. Galactose only differs in the position of the hydroxyl group on the fourth carbon. And fructose. All three of these have the same molecular formula. And that formula is C6H12O6. So they're isomers of one another. They're all simple sugars or monosaccharides. Saccharide means sugar or sweet. You can join these together to make, you can join two of these together to make sugars that have, carbohydrates that have two sugars. If you put glucose together with another glucose, you make maltose. If you put glucose with galactose, you make lactose, which is milk sugar. And if you put glucose with fructose, you make sucrose, which is table sugar. Whenever you have two sugars together, as in these three, we call these disaccharides. Di for two, saccharide for sugar or sweet, disaccharides. And this is all that we have to say about monosaccharides and disaccharides. On subsequent slides, we'll talk about polymers that can be formed from just glucose. We'll just look at glucose and see polymers that can be formed from this important sugar, simple sugar, glucose. Let's next make polymers of these sugars. Now, starting out, it won't matter which sugar we start with. Let's say, for example, we have a sugar here. Let's make it black. Simple sugar. Let's do this. Now we do understand that there's an O there, but I'm not going to draw that O. So this is just a sugar, and it's joined to, so that's a monosaccharide. It's joined to another sugar. That's a disaccharide. If you keep on making these, if it has quite a few, not many, but maybe 10 to 15, this sugar now becomes not a polysaccharide yet, but an oligosaccharide, because remember, oligo means a few. Oligo, a few, saccharide, sugar or sweet. So that's an oligosaccharide. And so for here, instead of putting an N there, let's just say we have a few of these. Okay. And so that's an oligosaccharide. If, however, let's just take this here. That'll save us some. Okay. Now let's do this. So instead of having a few here, let's say we have, we could put an N there, or we could say many, or we could just put an N here. So we have many now. And so now this is not an oligosaccharide now. In this case, if we have many, this is now a polysaccharide. This is a polysaccharide. So this is a polymer of simple sugars, and the monomer, of course, they would be sugars. The bond that holds sugars together, that bond, nope, I'm not gonna, you don't need to know that right now. Later on we'll learn what that bond is called. There are three polymers or three polysaccharides of glucose that are very important in nature. And on the next slide, we're going to talk about those polymers of glucose. But on this slide, we have our oligosaccharides and our polysaccharides. We've already seen our disaccharides and our monosaccharides. On this slide, we're going to look at some polymers of glucose. Glucose can exist in a variety of forms. The glucose polymers can exist in a variety of forms. Okay, let us look at the simplest one first. If you have, just gonna, this hexagon here will represent glucose for us here. So here we have glucose. Now remember, on glucose, the OH can go either up, on the one carbon can go up or down. So let's draw the OH here going down. So the O is going to join with another glucose. And then it's going to go down. It's going to join with another glucose. Okay. Now if this continues on. Okay, and let's say here that we have quite a few of these N. So these are going to be polymers. So we're going to have many of these, many of these. And it's one long string. This is a polymer of glucose, and then of course it is a polysaccharide. But this polysaccharide has a special name. It's found in plants, and we call this starch. We call this polymer of glucose starch. When the OH on the one carbon goes down, we call that polymer starch. And as you can remember, for glucose itself, the four carbon, the OH on the 4 carbon is already down, so this is going to be starch. Let's draw another one. I'm going to actually take this one because the next one is based on this one. So we're going to take here. Okay, I'm going to make a copy of this. Now this is starch. Okay. Now, what can happen sometimes, I don't know where I'm going to put this branch here. I'm going to actually move this. I'm going to move this over. I'm going to let that on. Let's get this by itself. Okay, here we are. Sometimes we can get a branch actually I can just use this again now that I think about it. So what I'm going to do, I'm going to make another, well, not a copy of this, I'm going to move this. Okay, made myself a copy of this one. I'm going to put it right here. And so what's going to happen, okay, I'm going to put it here. Sometimes we can make a branch. If you remember, this was a okay, there we are. Here we had a CH2OH was there at first, but now the O is going to be here and join here. And so this is the 1 carbon, and that's the 6 carbon. And so we can make branches off of this starch molecule. And so now it's not starch, because starch is found in plants. However, this branched starch molecule, this is found in the muscles of animals. And we call this molecule, we call it glycogen. And again, in both sources, and in both of these, we use these glucose polymers as a source of energy. We can eat potatoes and get energy from the starch, or we can eat, or animals can use we can use the glycogen that's stored in our own muscles for energy. And so these are 2 polymers of glucose that we can digest, that we can get energy from. On the next slide, we're going to look at an example of glucose in which these 1 to the 4 bonds, the O does not go down, but it goes up, and we can't digest those, and we'll look at those on the next slide. On this slide, we'll look at our last example of a polymer of glucose. Okay. Now, in this polymer of glucose, okay, we're going to draw our glucose molecule here. There's glucose, and we'll draw another glucose molecule, glucose. But now in this example, the O does not go down, but it goes up. Okay. This one goes up. And this one goes up. And of course, as before, we have many of these. So we put here. Okay, and we have many of these here. We'll put an N here. And so this is a polymer of glucose in which the O's go up. The 1,4 linkages go up. And so this polymer of glucose is called cellulose. Now, cellulose is a form of glucose, is a polymer of glucose that we cannot digest. We cannot digest glucose because we can't break these bonds that go up. So you may think, well, you may think that's not useful for us. But cellulose is found in things like, it's found in wood, which we can't eat, cotton, paper, and as you can surmise here, these are all things that we can't eat. So why are we talking about cellulose in a course about living organisms? One reason is sometimes in your body you have, of course, you have your stomach here, and then you have your intestines. Sometimes you'll have material in your intestines, but there's nothing really behind it to push it through, to push it on through. You eat things, some of the foods you have cellulose in them, and we call this roughage or fiber. And so this fiber comes in and it goes and it pushes things through your intestines. It kind of keep your intestines clean. And so this is fiber, dietary fiber, and that's good for us. And so some of the foods that we eat have cellulose in them, and this cellulose provides dietary fiber. And that's why cellulose is important in physiology.